Welcome, Voyagers, to the Epigenome control room aboard the Stellar Biome Station. I'm Kilaim Bey, your cosmic bioarchitect. For millennia, humanity believed destiny was written in stars, overseen by supernatural force or zodiac signs. But what if the thread we call fate waves not only through the fixed bricks of our DNA, but through environmental furnaces, living causes across time? Tonight, we embark on epigenetics. Are our genes our fate? An odyssey where base pairs remain constant, yet gene expression soars on wings of chemical marks. Forget astrology. I hate it. I don't believe it, by the way. So forget astrology. Here, carbon methyl groups, histone cups, and environmental triggers write the script. This is no dry lecture. It's sci-fi changed, yet rooted in cutting-edge molecular biology. Stay tuned for code brainwaves. To unlock my Udemy course, SAT Biology 101, Unlocking Life's Blueprint Level 1, you'll receive high production 3D molecular demos and 3D printable epigenetic models. Subscribe and comment. Your support propel our galactic research, collaborating with alien intelligences beyond. Fasten your belts, lower your vocal register, and fire our nanoshuttle's engines deep into the epigenomic layers. First, let's define epigenetics, the chemistry above the genome. The ATGC letters you're memorized in high school stay put, but which genes ignite or dim depends on chemical marks. Our cellular control panels. First of all, the gene expression dashboard. Every gene connects to a dashboard inside each cell. Epigenetic marks act like light switches, dictating where RNA polymerase can land. Example, the identical DNA sequence becomes a neurotransmitter factor in brain neurons, but a detox workshop in liver cells. The same code, divergent destinies. The core mechanism, methylation and histone modification. DNA methylation, when a methyl group CH3 tags a cytosine base. It slum-shut gene expression sometimes for decades. Histone acetylation. Acetyl groups in histone tails loosen DNA's warp, remove them. Chromatin tightens, transcription halts. Epigenetic inheritance. Nutrition, stress, toxins in the womb can program adult metabolic disease and behavioral quirks. In mice, specific histone marks pass across generations. I'm talking about a story. Babies born during the 1944 Dutch famine later suffered heart disease and diabetes. Maternal starvation penned chapter 1 of an epigenetic saga. Thus, to claim genes are destiny is to ignore epigenetic canvases and environmental forces shaping our fate. Now, plunge into nano detail. The chemical engineering being DNA methylation and histone tail modifications. DNA methyltransferase, DNMT. DNA methyltransferase, DNMT family. DNMT3A, 3B establish new methyl marks. DNMT1 propagates mark during cell division, like a digital backup system in a Starship core to prevent data loss. Target zones. CPG islands, clusters of CG pairs near promoters. A methyl flag here erects an immovable barrier to transcription machinery. Histone acetyltransferase, HATS, and deacetylases, HDACS. HATS add acetyl groups, neutralizing histones positive charge, losing DNA histone grip, chromatin opens, genes on. HDACS remove acetyl, chromatin closes, genes off, impact memory formation. Neurological disease rely on this off switch. Additional marks about methylation, phosphorylation, ubiquitination. Histone residues can carry methyl, phosphate, or ubiquitin groups, expanding expression possibilities like tweaking chords in a cosmic symphony. A single note shift changes the melody entirely. Imagine chemical toggles that compose molecular novels, writing cellular fate one mark at a time. The real marvel, environmental stimuli, writing epigenetic scripts, not as supernova's radiation, but diet, exercise, toxins, stress. They echoes across billions of cells. Nutrition and metabolic signals. Methyl donors, folate, B12, methionine shape, DNA methylation. Example, vegetarian mice exhibit gene expression patterns resisting obesity. Thanks to altered methyl pools, stress and hormonal waves, cortisol surge, trigger histone modifications in hippocampal genes, altering memory and emotion pathways. case, offspring of the World War II veterans showed heritable methylation changes on stress-responsive genes, toxic exposures, air pollution, heavy metals, endocrine disruptors, leave indelible marks on fetal epigenomes, shaping immunity, metabolism, and hormone balance. I researched babies born after Fukushima showed aberrant methylation in metabolic genes, physical activity, and lifestyle. Exercise sparks histone modifications that upregulate anti-inflammatory genes. Health living is an epigenetic manifesto. Daily choices are brush strokes painting a dynamic portrait atop our genetic blueprint. Genetics isn't just academic, it's revolutionizing medicine, agriculture, or even space travel. Disease, diagnosis, and treatment. Biomarkers monitor prompter methylation in blood, urine, or salvia to detect cancer and neurodegenerative disease early. Therapeutics, HDAC and DNMT inhibitors in cancer aim to erase pathological marks and reset cells to normalcy. Agriculture and food security. Epigenetic engineering crafts drought-tolerant, nutrient-rich crops, crucial of Martian greenhouses. Aging and regeneration. Induced pluripotent stem cells, iPSCs, use epigenetic reprogramming to rewind time, generating youthful cell profiles. Spaceflight and human adaptation. Microgravity and cosmic radiation rewire the epigenome. Future astronauts may use epigenetic drugs or training regimes to shield against these changes. Genetic armor for the cosmos. Epigenetics guides the hand that sketched atop our genetic script. Here, true miracles take shape. Our exploration through epigenetics, are our genes, our fate, concludes. I hope you now see DNA not as destiny's cage, but as the foundation for a dynamic, interactive masterpiece. Coupon code BrainVase. Use this on Udemy for SAT Biology 101 Unlocking Life's Blueprint Level 1. Download the 3D printable epigenetic model and bring molecular science to your tabletop. Subscribe, leave your thoughts, and prepare for our next voyage. Synthetic life, artificial cells, and new biological worlds. We'll chart nano-evolution's frontiers. Don't miss the journey.